Guys, welcome back to my lectures. In today's lecture, we will see about the transposons, that is mobile DNA element. क्या आप लोगों को पता है कि अपने ह्यूमन जीनोम में ऐसे डीएनए एलिमेंट्स होते हैं जो कि जंप कर सकते हैं, जो कि मूव कर सकते हैं. यस, दे आर कॉल्ड एज ट्रांसपोजॉन्स. So transposons are also known as jumping genes, or you can say them as movable DNA elements. They are present mostly in prokaryotes as well as in eukaryotes. It was first discovered by Barbara M.C. Clintock when she was studying cytogenetics in maize plants. Thus, transposons are most probably found in 80% in maize plant, while as 10% found in fishes, and as well as it can be found in human genome as 45%. Transposons can cause various genetic alterations based on a process of transposition, that is insertions, deletions, duplications, translocation or excision in the site of integration. That means transposons can do transposition, that means one DNA element can move from one place to another place, or you can say one DNA element or any DNA sequence can move from one genome to another genome via insertion, that means directly inserted, or deletion, that means some DNA sequence can delete, duplications, that means DNA sequence can gets duplicated, translocation, that means one gene sequence can translocate or move from one place to another place, or excision, that means cutting. Okay, by using all these mechanisms, transposons can do transposition and thus can cause various genetic alteration. Now let's see the characteristics of transposons. Transposons can modify gene structure, thus can cause gene expression. That means some types of genes can be expressed, or we can see genes are expressed in some type of individual or any types of plants or bacterial species due to presence of transposons, because they can move from one place to another place, thus can cause genes, thus can modify gene structure. It can create novel genes because it can do gene shuffling, as I have said earlier that it can do insertions, deletions, right? Thus can do gene shuffling, that means gene की position change हो जाती है, gene shuffling हो जाती है. Transposon can cause diseases and thus can cause mutation. That means mutations, what do you mean by mutations? Mutations are nothing but the sudden changes in the DNA sequence or gene sequence. Thus transposons are responsible for causing mutations. Transposons most of the time carries antimicrobial resistance genes inside it. We will see in the structure where you can see transposons can carry antimicrobial resistance genes inside its DNA sequence. Okay, because transposons are nothing but the DNA element and thus can have different types of genes or any types of sequences, so it also have antimicrobial resistance genes. It can move during transformation, transduction and conjugation. We have seen already what is meant by transformation, transduction and conjugation. So during this genetic transfer process, transposons can move from one place to another place. Transposons most of the time code for enzymes which are responsible for self-replication as well as DNA insertion in host chromosome. So these were the characteristics of transposons. Now let's see different types of transposons. Transposons are characterized into various types. Some are of showing here. First one is the simple transposons, or also known as insertional sequences. Second is the MITs, that means miniature inverted transposable elements. Third is the TN or complex transposons, and the fourth one is the retro transposons, or which are responsible for doing retro transposition via using RNA intermediate. We will see in the detail in our next videos. In today's video we are going to see only simple transposons. What are these and how it gets transfer from one genome to another genome. Okay, so you can see in the diagram, simple transposons, also known as insertional sequences, the name itself says that it can get inserted into any type of DNA element in the host chromosome. Okay, so they are simple transposons are also called as class 2 or insertional sequences or insertion sequence. Okay, they are nothing but the DNA sequences and consist only of DNA element. It only have DNA element. Okay, these transposition elements have shorter sequences which are in very shorter length. Okay, that means you can say 800 to 1000 or 1500 base pairs can be seen in insertion sequences or simple transposons, you can say. They do not code for proteins. Okay, they are present in many bacteria as well as they are also found in bacteriophages also. They move from one place to another place via transposition using the mechanism that is cut and paste mechanism. That means the required DNA element which is get to be transfer or transposed from one place to another place is get कट. मतलब जो DNA एलिमेंट हमें मूव करना है वो अपने आप कट हो जाता है और जहाँ पे इंसर्ट होना है वहाँ जाके वो पेस्ट होता है. ओके, सो दिस इज़ नथिंग बट द रैंडम एंड natural process present in the bacteria. We do not do it by artificially. So via transposition, simple transposons can move from one place to another place by cut and paste mechanism. For this transposition they require transposase enzyme. Okay, you can see in the diagram. Let's discuss using diagram. So you can see these are the target sequence. That means here you have to insert these simple transposons or insertion sequences. So simple transposons first of all have the ends, that means having inverted repeats, or you can say IR. Inverted repeats are present at both the end of insertion sequences. Okay, and this is the whole transpose. This is the whole transposon. Okay, so here the transposase enzyme which is encoded by this DNA element will bind to this inverted repeats, and using cut and paste mechanism this insertion sequence will get inserted into the target sequence, and using the Watson and Crick method, using the DNA ligase enzyme, the cut DNA will be gets ligated and the new genome will be formed. Okay, so in this way transposition happen in simple transposon. In our next videos we will see in detail about 3 different types of transposon, that is MITs, complex transposons or composite transposons, and retrotransposons. Thanks for watching.